So I'm here today to tell you about the biggest parasitic disease that you have probably never heard of. Visceral leishmaniasis is the second biggest parasitic killer worldwide, right behind malaria, and currently has very limited treatment options. It's caused by the parasite leishmania, which is able to get inside of a host's white blood cells after they're bit by an infected sandfly. Now, cells talk to each other in a lot of different ways. One of the ways they do this is by trading around exosomes. Exosomes are small packets of information which contain proteins as well as other molecules that are able to tell other cells about the state of the cell that sent them out. Most cells do this, including the white blood cells that those parasites inhabit. Now, we know that some diseases, the germ that gets into the cell, it's able to hijack the information that's sent in these exosomes, sending misinformation and causing the disease to become worse. So what we want to know is this happening in VL? So the way I've looked at this is by taking blood cells from healthy human donors and infecting half of them with these parasites. Once they're infected, I collect exosomes from them and then use a technique called mass spectrometry to open them up and read what's inside. What I have found is that the exosomes from the infected cells do contain different information. They contain fewer proteins that we would expect to see in a cell that is functionally finding and killing parasites, and more proteins that are related to how lipids, which make up fats and steroids in the body, are processed. Now, this last is particularly interesting to us because we know that one of the things that causes fatalities in VL, it's mismanagement of lipids leading to liver failure. So why does this matter? Why do we care? VL, as I said, is the second biggest parasitic killer worldwide. Not only that, but fatalities are rising this decade. There is no vaccine, and currently the only treatments available are chemotherapies, which are not only toxic to patients, but expensive, difficult to administer, and there's rising resistance to them among parasites. We need better treatment options. And to find them, we have to know more about how this parasite is making the body sick. For exosomes to be a pathway, they'd have to be different. And we've now seen that they are. So this opens exciting new possibilities to interrupt how the parasite is making us sick and improve patients' outcomes. Thank you.